What this is about is, I title it Translational Ongoings. And since I know a little bit about the Vigil stuff, one of the things that I thought has always been a mystery to all of us is, when we're doing our translational work, particularly in phase 1 studies, we're harvesting samples, we're, processing them in specific and unique ways, and it's a critical component in defining, mechanism of action, pharmacokinetics, things like this. So I thought it might be fun to actually go into all that goes into the development of the assays, how they're relevant, and what they lead to for clinical trials. And it's more difficult oftentimes to understand immunotherapies and the translational aspects of an immunotherapy approach. So I was going to take you through some of the aspects and development issues associated with the Vigil product. I think many of you guys know what this is. It's a targus tumor vaccine that is transfected. That means the DNA of a GM-CSF DNA and of a TGF-beta knockdown DNA is placed inside tumor cells, and the purpose of it is to show tumor antigen and knock down immune inhibitors that cancer cells produce and make large amounts of immune stimulators. That's what the GM-CSF is. And so if you go into any technology that comes into our clinic, there is a long process that has, unveiled to the point that enables that to fulfill IND requirements, go through the FDA, and literally become a clinical trial that gets offered up to us to use for patient management, okay? And so I thought in giving you some background on things that went into many people's heads that thought, would this be something that might be a step forward? And so there are many products that were tested years ago. We find that a limit to it. We found that some activity but not enough, or we find some toxicity, and we're trying to get around the toxicity. So a lot of product development involves learning experiences from prior products and improvements and tweaks to them that try to reduce the toxicity or improve the efficacy. And so on this first slide is a picture of patients, all who were treated at Mary Crowley, all who received immunotherapies, and virtually the same mechanism was involved in each one of these. The first one you guys know well, although some of you guys may not, but that's Grover, and he received a injection of interferon expressive tumor cells, and that led to a significant response of a very refractory melanoma that literally had gotten to his brain. And based on his response, he was able to survive 18 years without recurrent disease, and he basically lived his normal lifespan. And shown on the picture to the right is his daughter, who was getting married at the time he received his therapy, and her child, who's in front of him, who he saw her aged 18 years old. And it was, that's our idea on what we want from development of new technologies. And so from that, we thought, well, is there a better way to improve the stimulation we found with gamma interferon? And that's where the GM-CSF part came in. And some of you guys know her, many of you don't. But the second picture there, patient 01 of the trial with GVAX, was Connie West. And Connie had multiple lesions in her lungs of lung cancer. She had failed all standard therapy, was given a prognosis of about 7-8 months to live. And she also is still alive now 13 years later. This shows the response before and after to what we thought was going to be an improved immunomodulatory therapy. TVAC was used in someone who is a senior member of our clinic, her father, and he had a dramatic response as well and lived 5 extra years. Eventually he did recur. So we realized we didn't find the answer with this approach, but the GM-CSF part of this was delivered by a replicating virus. The intent was to try to expand the GM-CSF that was delivered to the cancer cells. We know when GM-CSF turns on the immune system that there are inhibitors that cancer cells make, and on the left lower case is another patient with a dramatic response who received knockdown of TGF-beta. That patient also had significant response, lived many years longer, but the dominant TGF-beta is TGF-beta 1, not TGF-beta 2. A subsequent study was done where we combined the activator with an inhibitor. that study was also associated with a dramatic response. And as you can see, though, in looking at all patients, it still wasn't enough. And then the most recent step, is this a stepping stone to something in the future or possibly something that can secure FDA approval? We don't know, but that's where the Vigil comes in. But this is the young woman, the 18-year-old, this is Carly. And Shannon's up here, and she's still alive with no disease, finished her school, and we recently got acceptance of her paper. So, so again, you can see the progression of response to various immune modulators, but each one subsequently attempting to improve the next one, improve the next one, improve the next one. That's 18 years of work. We're still not there yet, but we found many improvements. So, you know, a question is, something that we've been trying to answer, where I think as I go along, you'll see that we're getting some answers now, is what's the mechanism of this durable complete response in these 6 patients, but in many others that I haven't shown on this slide because this isn't room. And so on the next slide, you know, our most important point isn't to cause the cancer to shrink. That's certainly a key point. But if we can improve survival, particularly improve survival without associated side effects, then we're doing something more for the patient. If we can improve survival significantly without the cancer being there, that's even more. And so that's where we're headed. And that's why you'll see in many patients we manage in the clinics, not only will we offer the technology, watch the response, but if we have a chance to come in there surgically, come in there with radiation to debulk disease where we can't see it, we take advantage of that. Our end goal in our clinic is to get them down to where we have no visible disease and durable survival without cancer-related pain or treatment-related toxicity. So on this slide, you can see with the Vigil technology, 80 patients who went on to the phase 1 trial, the expected survival of these patients is where the red line is. It's really 6 to 8 months. So you can see there are a number of bars beyond that red line, and each day the survival is getting better. All those patients with the arrows are still alive. Now, these are all patients who had failed standard of care, who had no options after second-line therapy, and are still doing reasonably well. So that's an encouraging thing, but why are some of these patients living so long? What's the mechanism of this? And so that's the ongoings of translational research. It's one thing to give the therapy, follow the patient, watch for side effects, try to find the best dose and schedule, but it's another thing on understanding what's going on in the immune system and understanding what of the immune system can we modify? How can we make that patient less resistant and more sensitive, okay? And as we now know what the human genome expresses, we can track various genes, we know the cells that make up the immune system, we're starting to track those things. So the blood tests that you guys do when you spin down the samples, when you harvest the samples, all these blood tests for the patients, a lot of this goes into identifying specific subset populations that we can track, understand what's happening with them on this therapy, and if we know that, we're either there, we got it, or we know how to tweak it to make it better. And that's what's so important about harvesting the blood samples, and what's so important about our clinic, the way it's set up, and the harvesting of the blood samples. We don't like to just draw blood on patients, but it really does go to useful assessments. So on this slide, slide 4, this is an assessment of what the cancer cell deals with when it gets into its niche. When the cancer cell develops in the body, it starts developing right where the genetic mutations occur. If it's in the lung, it's in a lung cancer area. If it's in the liver, a liver area. And it eventually creates an environment where it has a stable protective area. It produces inhibitors of the immune system. The immune system can't get in there. It'll oftentimes eradicate immune cells that do get in there, and it produces other factors that basically stimulate its own proliferative growth. And so there are really 5 basic mechanisms. If you look on the slide here, and again, this is relevant to the Vigil technology, any technology goes through the same thought process. So any immunotherapy that you have is going through the same process as Vigil, because these are the questions the FDA asks, these are the questions any doc, nurse, any patient asks. Well, why is this working? Why is this not working? And how can we make it better? And so technically with the immune system, there are unique markers that cancer cells have that normal cells don't have. And they get expressed by a mechanism called MHC. And when MHC is expressing the cancer antigen, the immune system can come by and grab that antigen and then take it back to lymph nodes and create new immune cells that can attack it. Well, if the cancer cells can inhibit the MHC from being expressed, it'll be able to hide its markers. So Vigil is trying to address that in that it puts in GM-CSF that forces the autologous tumor that's harvested to make that product to express that tumor marker. So now the immune system can see it. But the cancer has a lot of ways of avoiding the immune system. Even if it's expressing it, it can produce molecules called FAS and TRAIL that basically block the immune cell. They basically kill immune cells that come to the area, so they now are no longer able to function. This therapy doesn't deal with that. If the therapy fails, then that would be a logical combination technology approach. Block FAS, block TRAIL, because that's not addressed by this therapy. So you want to know the good and the bad of any therapy. Or what is it, the ugly and the Clint Eastwood movie, the ugly, good, bad, and ugly? Yeah, you want to know that. So then there are tumor suppressive effects. TGF-beta is the most dominant TGF suppressive molecule that tumor cells produce. When TGF-beta is in the area and the immune cells come to the area, they just shut those immune cells down. And they're like, can't do anything. APCs, those are the monocytes and the macrophages that come to the area. Those cells grab the tumor markers from the MHC area and they show them to immune cells so they can generate receptors that can now bind to the cancer. And when they bind to the cancer, they'll release gamma interferon, they release toxins against the cancer. So you want these as optimally functional as you can. So the Vigil technology will address 3 of these points, but there's 2 points that it doesn't touch at all. And those 2 points, the FAS and the checkpoints, may prevent a good response in some patients. So if you know that, you can have a strategy of how is it by itself as single agent, but then what can you do to improve it? Can you modify the product itself, or can you actually look at combination studies? And as you guys know, there are a ton of combination checkpoint inhibitor studies going on now because very few therapies address what the checkpoints address, and that is a big inhibitory part of the tumor.
Weren't you guys adding Crombo to Keytruda into the Vigil?
Yeah.
Is that in an ovarian setting?
There's an ovarian trial, there's a breast trial, there's a melanoma, there's a lung. It's not open yet in ovarian, but the ovarian patients that get onto the phase 3 trial, many of them don't qualify, about 10% of them. So now there's about 30 patients who have vaccine made, but there's no options for them. They've gone through second-line treatment now, so why not offer this in combo to learn the effects? So that is going on. So the main aspect that the cancer cell is trying to protect, the main way that the immune system finds the cancer is its antigen on the surface. And cancers have thousands of antigens on the surface. But sometimes when the immune cells bind to other antigens, they don't turn on a reaction. It's the driver neoantigen that causes the response. So that's the holy grail of the cancer cell that they want to hide to not allow the immune system to kill it, basically. And so that's what all this mechanism is about. It's about hiding the driver neoantigen. So basically, that's how the immune system protects itself. The mechanism for the product that we're talking about, the Vigil product on the next slide, is one where the cancer cells have a plasmid put inside them. These are cancer cells that are harvested from the patient before starting treatment. The plasmid is put in as part of the manufacturing process. A technician goes to the OR to secure that tissue, and it's handled in a special way to keep it sterile and then expose the plasmid to tumor cells. That's just an overnight incubation. It goes in via electroporation. And the electrical current is put through the cancer cells, pores open up, and then the plasmid goes inside. And then it starts making the GM-CSF gene and the bifunctional furin that knocks down the TGF-beta by knocking down furin, and it basically provides the functions that we're trying to turn on, an enhanced function to the macrophages and a high expression of the MHC to tumor antigen, and it takes away the tumor inhibitor that the cancer cells get.
How long does that take?
24 hours. So to make it takes 24 hours, and then about 4 days after it's made, the GM-CSF product, which cancer cells don't make because they don't want to stimulate the immune system, and the TGF-beta, which they make gobs of, is measured. And what is shown as part of product release before it comes to Crowley to give to the patient is a high level of GM-CSF, over 30 picograms per 10 to 6 cells per ml, and a marked reduction in the TGF-beta, on the average 93% reduced. So now you can see the suppressive effect is markedly reduced, the stimulatory effect is markedly elevated. Cancer cells don't want that. It's sort of taking the wall away from the castle. The other part, by showing the MHC, when this is injected into the dermal layer of the skin, lymphocytes will come to the area and they'll grab the antigens. The mononuclear cells will grab the antigens and they'll turn on the lymphocyte system so it can now find the tumor cells. So on the mechanism slide here, you see that cell with all the little U-shaped things on it. Well, normally, lymphocytes don't have any of those U-shaped things on them, and those U-shaped things, those are the receptors that are needed to bind the tumor antigen elsewhere in the body. So once you create the population of those little red dot cells with all the U-shaped things on them, they're loaded with those receptors. It just takes one little antigen for those things to find the tumor and then release the gamma interferon. So now they're more effective at basically finding the tumor and then more active at being able to kill the tumor. So that's what happens systemically following the local treatment in the skin. So it's injected intradermal. I don't know, who's doing, are you doing the injections on some of these?
Well, the nurses have been
doing them. Nurses are usually doing them?
Yeah.
So when they give the intradermal injection, that local effect creates the manufacturing of those sort of U-loaded cells, and those things get loaded up in the blood to go attack the tumor cells.
So this is when you're processing, you put that stuff in, what do you call it, in 24 hours it sits in there.
Yeah, the plasmid.
The plasmid, that's what I was thinking of.
Yeah.
So that is what caused the plasmid or whatever, helps break down furin, causing the knockdown, which causes the increase, the blockade of the TGF-beta.
Right. It decreases TGF-beta.
Right.
Yep.
And that's what causes that inhibitory effect once it goes into the cell.
Right. And so what this is attempting to do is, when that's injected into the skin, there's macrophages. The organ that is the richest in macrophages in our bodies is the skin. And so by injecting this intradermally, you have the highest ratio of macrophages to those injected cells. And 10 to the 7 cells are injected. One area, they're irradiated, so they can't grow, but they have the antigens on them, they express the antigens, they make the GM-CSF, and so the GM-CSF itself attracts the macrophages, so they come over there like crazy, and that's where you get the little dime to quarter size redness over the site. So those macrophages come into the site. And then it's a couple days later in the body that the actual cells get into circulation, okay?
I'm sorry to interrupt you. Clinically, have we correlated, I should maybe know the answer, I don't know. Have we correlated like the size of the rash with like an LA spot with a receptor?
Yeah, it hasn't correlated.
It hasn't, okay.
No. And that, in some studies, there's some suggestion the bigger rash, the better correlation. And maybe that we just haven't had a big enough rash yet. But part of the issue is once the macrophages get the antigen, they then travel to the nodes locally, and we change injection sites. So there's nodes all over the place. And so the action doesn't necessarily take place for the redness with this approach right there. It really takes place in your lymph nodes that are distributed by injections all over the body. And so you're sort of diluting the redness effect on purpose so you don't get a big red area. You know what I mean? So it's kind of interesting. The other thing that, at least in my brain and thinking of the technology, to me, we're smart at what we do, people, scientists, but in all honesty, we're not that smart. And so your body is real good at healing itself. If you cut your arm, that heals pretty good. Now, think about this. If we went into a cut and we decided we're going to figure out how to speed that repair process up, we're going to have to think about platelet function, endothelial cells, angiogenesis stimulator factors, skin regrowth, skin speed up. Now, if we try to break it apart and really figure out what therapy we should give to help that cut, think about how difficult, how complex that is. Well, the cancer cells is more complex than that. And we think we know what we're doing on how to attack the cancer cells. It's pretty hard. So in my mind, if we can assist the body to heal itself against cancer, that may be a better approach. And our body has great immunologic systems with the APC presentation of antigens, with MHC expression, with T-cell activation, CD8 cells, that why do we want to really mess with that? Let's figure a way of turning that on, but turning it on against the antigen target, and that's where the autologous tumor cells come from. Hey, Jim.
Hi.
So this is translational ongoings. And I'm using Vigil as the example. So I'm going into what are the factors that go into assay development, the blood tests are done, why it's done, and what it can lead to in a product development. And every single product that we develop here has had this process. They've had to think through, they had to develop the assays, they had to figure out how to process it, how to utilize it, how to optimize it, and then what they can do with it in terms of combination therapy or new product development or tweaks to the current product to make it better. So that's what we're going to run this. So this is the translational part. This is part of the deep dive. We know about the clinical part. This is really the translational. So the FDA, when we talked to them about this mechanism on the prior slide, the key thing that the FDA said, listen, if we look at this, I asked them, my words were, what would raise your eyebrows about immune assessments to allow us to move this product forward more rapidly? What sort of proof of product activity would you feel would be, significant? And they said, well, if you look at all this, product release, we get the GM-CSF. We trust GM-CSF will do what it does. We get the TGF-beta knockdown. We trust you're going to be able to demonstrate that. We're going to require that as part of the product release. We know you can't figure out what the antigen is, but the real thing that we want to know about isn't about the tumor and the clearance where you're injecting it. We want to know what happens to the metastatic disease, and what is it that gets into the blood that is supposed to go to the metastatic disease sites and clear those tumors, or at least control them, okay? So they targeted this cell, the little cell with the U-shaped things, as what they want to know about as evidence of product activity in the patient. They wanted a functional assay is what it's called. So we thought long and hard about what kind of assay should we develop that would be a functional assay. How can we measure that cell? This was not developed. And we decided that on this slide here, slide 6, we could take tumor cells. We're harvesting them to make the vaccine. We could take a little aliquot those cells and take the cells that haven't been transfected, because the tumor cells in the patient that are in the other parts of the body, they haven't had the GM-CSF gene put in them. They haven't had the TGF-beta gene put into them. So this therapy has to work against those cells, okay? Not just the ones that are transfected. We know that they're going to clear when we inject them. And so a assay was developed where a antibody was placed on the bottom of the plate. The antibody was there to grab gamma interferon. The main toxin, when those squiggle cells attach to the known neoantigen in the tumor, that induces a reaction among those cells, and the reaction is release of gamma interferon. That's a toxin that cancer cells hate. They die when the gamma interferon gets released. If we could measure the gamma interferon, that would be evidence of that reaction taking place in the test tube. Okay, and that's called the lespot assay. And so we took the tumor cells, placed them on a layer above those antibodies. Yeah, it's a porous media, but tumor cells stayed above, the antibodies below. And then we put the mononuclear cells in that mix at a specific number, a 3-to-1 ratio of mononuclear cells to the tumor cells, and the tumor cells should have their antigen. It should be expressed with MHC, but not much of it. These are the tumor cells that haven't been transfected. So they should be expressing the antigen similar to what's in the body at the other disease sites. So can these cells that we're saying is in the blood, where we had no idea if they really were or weren't when we tested the first patients, will these cells come out? Can we see evidence of them? And will gamma interferon be released? So this assay is designed to assess that. And the assay is used to check peptides. MUC1, you've heard of MUC1, on 10 is the MUC1 related. So this assay was done for those studies, for many vaccine studies that you can think of several of them in melanoma studies that we've done. Typically when the assay is done, you'll take a certain number of those peptides and you'll take mononuclear cells. Well, typically you can't see reactivity unless you have at least 1 million mononuclear cells in the assay. It takes 1 million cells to just get a few to respond. And if you get more than 10 of them to respond, that's considered highly significant, okay? 10 out of 1 million. Well, when the assay was tested on the first couple of patients, it overwhelmed the plates. So we had to downsize the number of mononuclear cells that we placed to cause a reaction. And the number that was felt to be where it's not too many reactions where you can't count the plate, but it's enough to where you can see the reactions, was 10 to the third. So technically 5 times 10 to the third autologous blood cells. And then there's a set number of autologous tumor cells that react against. So that's the lespot, the spot part of it. It's a red reaction. When the interferon binds the antibody, it'll cause a red reaction. It's a little more complicated than that, but technically that's what it is. And so 200 spots are seen out of 5 times 10 to the third cells. That's considered way more. Now, it's not simple.
Color something we just overlay on it for the
yes. Yeah.
You used to talk about purple. Now it's red.
So
it's just something we...
Did I just say red?
Yeah.
Oh, okay. Yeah, because it looks kind of, it's red between red and purple, but it's the same. Technically, there's the chromogen that gets added is the color reactant, and so actually at Mount Sinai, where this is verified, it's blue. So, it depends on, yeah. So, so one of the things you got to make sure of is do you have the tumor cells? Do you have the peripheral blood cells? And so technically the peripheral blood fraction is CD8 cells. And so shown here are the verification of the tumor cells. You can see the yellow arrows and red arrows. One thing that happens is when you take cells, you freeze them, and you thaw them to run the assay, is you lose some cells. Not all cells survive the freezing process. And so we have to know, what proportion of cells are still alive and viable, what proportion have died off, and it's a manual count. So we're now working with a Mount Sinai to do an automated flow cytometry mechanism that'll do the same thing. But in the lespot assay, the manual count is shown here. Podmo does the readings. We also have to make sure that the lymphocytes are healthy and they can survive. We have thawed lymphocytes out 6 years after freezing them for storage, and they've been very functional and healthy. So it looks like that the process works, so you stick with that. And but these are the cells that are harvested in the clinic here. They're centrifuged at Gridalis, and then they're stored as pure mononuclear cells, and then brought out for the assay to be run. This just shows on slide 9 the complexity of an individual patient. You have to run them all at the same time. There's a lot of variables. The assay is not entirely quantitative, and so shown here are your negative control. Obviously, if you don't have the tumor cells there, do you get any reactivity? If you do, you get a problem. So you can see on this patient there's no reactivity at all to blood cells, but no tumor cells. Then you have a antigen works on all peripheral blood cells. You got to make sure the peripheral blood cells are reactive. Are they not reacting because they just are dead? And so on the second 3 tier of the plates, that's PMA, and all lymphocytes will react to that. You can see that's very robust. So the lymphocytes are working well, and there's no reactivity. They're not overworking because there's no reactivity on blind plates. So then you go and take the patient's own tumor that has not been transfected, and you take the tumor that's been transfected. If you take the transfected tumor and you don't see reactivity, you're probably wondering, well, is your therapy really going to work? And then if you take the non-transfected tumor, you're probably wondering if your therapy is going to reach the tumor sites at distal places. So you want to see both of them pretty positive, and the expectation would be that the transfected ones are probably a little bit better because they don't have the TGF-beta. And it turns out that's typically what is seen. So this just shows various time points. Baseline, which is pre-treatment, where you can see on the 3rd line down, there's no reactivity, 0 to 1. But at month 2, month 3, and subsequent time points, end of treatment, and then months after treatment was stopped, we're seeing the reactivity. You get the assay now? So when we draw these blood tests and send them over to Gridalis, this is what's being done with them. And so I'm going to try to show you on what it leads to. Okay.
When you said that we can't do just one at a time, so that's why they're done in batches? Is that what you're
This is done in patient-specific batches, so that since there's variability, each time you run the assay, you have 10 to 15 counts. If you run this patient's baseline sample, one time, and when they're month 2 another time, when they're month 4 another time, it's harder to compare just for that patient. So when you run it all at once, if there is variability, it's been always to that sample. But this patient comparability, it looks like this patient clearly increased at month 2, and then relatively maintained it, relatively stable after that. So bumped up and then stable.
So it's all time points for a single patient.
Yeah. But there's definitely bouncing around, and it's been determined that it's not quantitative enough to where you can say a high one means it's good, a low one means it's bad. It's above 10. Obviously, we're more comfortable it's way above 10, but that doesn't necessarily mean that it's better, 150 versus 50, so to say. And so here's more important correlation. You like to see the mechanism proven by the lespot reactivity. You like to see these cells in circulation, but why do you like that? You like that because you're hopeful that it'll correlate with disease stabilization, ideally disease response, and ideally no recurrence, okay? Well, the start is stabilization. So these are the first 30 phase 1 patients. We track their survival. We track their lespot response. You can see 22 of the 30 had a positive reaction, and you can see those who had the positive reaction lived and are still living a mean and median of around 30 months. If they didn't have positive reaction, they only live, 11 to 13 months. That's still a little better than what you would have expected, but, which you want to be in. So we don't know why those that didn't react. We know they were all heavily pretreated with chemotherapy. It may have something to do with that, but that didn't come out as a statistically significant variable.
Is there a reason that 30 and it was N of 80 for the phase 1 trial? Like which 30 of the 80 were captured here?
The first consecutive 30. So we didn't run.
I get more data.
Right. After 30, patients 31 to 80 haven't been run yet.
Got it.
And the reason I shut it down is I understood this is good enough for FDA. We verified mechanism. We know what we're doing. So we could have run it to kill it. We may find some correlations if we do that, but there's limited samples. And could there be other studies that we could run on these limited samples? We've already proven ELISpot. We already know ELISpot is never going to be good enough to be a perfect quantitative. So are there other methods we could use and compare it to? So exactly what I'll go into. But just, yeah.
Oh, I'm sorry.
It's okay. You did mean to cut me off, but that's okay.
No, I was thinking out loud. So it was just, you had mentioned just a minute ago, you weren't sure why, like you would see some people's responses and they have like a longer response where some people might not respond. Their ELISpot might be good, but they don't respond, or those kind of things. I wonder, do you think it could be related to like MHC variability, like due to like ethnicity or race or
Yep.
what have you? I
Yep.
mean, and how do you figure that out?
Yep, I'll go into exactly that. And that is part of the reason for the variability is there are other factors that some we don't know about, some we do. We didn't account for because we didn't know about it at the time. It's been learned since. One of the classic ones is JAK expression. When JAK is mutated, JAK gene and the signaling of JAK is actually responsible for gamma interferon expression, gamma interferon receptor expression on the tumor cell. So if the tumor cell does have gamma interferon receptor, the gamma interferon that's produced by the mononuclear cells doesn't do anything to it. And so one of the protective measures of tumor cells is to have a JAK mutation. And so obviously a patient with a JAK mutation likely is not going to respond very well to this. Now, it wasn't designed in the study to look at the genomics of all these patients, but that's some of the stuff we're going to start looking into now. Did this patient have JAK mutation we didn't expect? And there's other genes that are similar kind of mechanisms. And there's more than one gene on the JAK signaling pathway, not just JAK. So yeah. The other thing that you'll note, typically when you give something that stimulates something else, you typically see if you give it more, you'll stimulate it more. But in every single example of the ELISpot following long-term over all these patients, it bumps up pretty quick, and then it plateaus. Now, the patients' plateau seem to be at different levels. If you look on this curve right here, it's plateau high, plateau low, but it's plateaued. They're not continually going up, okay? It's very rare if that would happen. There's some variability in the assay where you get some up and down, but for the most part, bumps up, plateaus. So we think that something is being produced by the body to limit an overextended immune response. And that's going to come into play a little bit later as I tell you about it. So a lot of things are learned just from this little thing. If you look at the randomized trial in ovarian cancer, where there were 31 patients who received Vigil, there were 11 who did not. There was a relatively impressive, with a small number of patients, improvement in time to relapse, an improvement of about 400 days by the mean comparison. Again, the ELISpot was correlated to response, and all patients but one turned on with ELISpot. The ovarian patients were a little bit less heavily treated with chemotherapy, so we think the chemotherapy is a factor on the immune responsiveness. The other thing that was interesting, it was a crossover study design, if you remember. So 8 patients did cross over in the control group, and you can see that 7 out of 8, even though they were negative the entire time they were in the control arm, multiple time points, none of them were positive by ELISpot, not only at baseline, but also afterwards, but they didn't get anything afterwards. But when they did cross over, they did turn positive just like the other ones did. But it's kind of curious that the median level of the ELISpot was quite a bit higher in the patients treated with Vigil earlier than those treated with them later. Is that a factor? I don't know. It's not enough patients yet to prove that. There are only 11 in the control arm. But that was kind of curious. So then if you look at the various sub-studies that were done, there were studies involving Ewing sarcoma patients, like melanoma patients, colorectal patients who got concurrent FOLFOX chemotherapy, and in fact, low dose. One of the problems with this technology is you have to harvest tumor tissue, and you have to harvest, a large marble size of tumor tissue to make the vaccine. Well, not all patients have that. Sometimes there's dead cells in the tissue harvested, so you don't have enough viable cells to do it, so you get failed manufacturing because of the difficulty in getting enough tissue. So a 10-fold lower dose in a small group of patients was given in a small cohort of I think it was 18 patients, and 12 we did spots on. And of those 12, all 12 of them, with the 10-fold lower dose, actually turned on the spot. And if you look at their survival, it's not on here. It's very similar to what the other these are the phase 1 patients. The other phase 1 patients did on survival. So maybe that we can take a core biopsy and get sufficient tissue. That was the level of the low-dose patients. Basically, 1 or 2 core biopsies worth of tumor tissue. So you can see that would be more logical. Now, that's not being done in the randomized trials right now, but that may be something to be a future opportunity, especially thinking of breast patients.
Get access to
exactly.
Create vaccines.
Exactly. So there's a number of publications. You can see these and grab them. So you get how assay was worked out, everything along the process, the harvest of the blood cells, the harvest of the tumor cells, maintain the viability, rapid response, reagents you react with, contamination, all these things are like, whoa. But in the end, you can see the results have been produced, and they've been relatively consistent. That argues to really good performance in the clinic, and also, good management in the team managing the samples to get these results. You know what I mean? So is that it? I mean, where does the mechanism of action, that's really what this is proving, where does it take us? It certainly justifies this technology. It was a purpose of the IND in the phase 1 IND, and it justifies moving forward to phase 2, phase 3 type trials based on the mechanism. The FDA is very satisfied with these results, and it's supportive of further product advancement. So that's nice, but is that it? Well, in my mind, it's not. We can learn a lot more from this, and that's the second part of what's worth going into on this, and this is sort of part of the ongoings of translational. So mechanism of action can be a basis for new product development, strategic combination. If we find out what is it that doesn't allow this spot to continue to go up, is there something else we could do about that? We got a certain level of these cells in the blood. Well, can we supplement that by giving them more of these cells? How can we give more of these cells? Is there some resistance factors like JAK? Well, then we shouldn't put JAK patients on it. Why waste their time? They should be on some other therapy that's more likely to be better. So those are things that you can look into for that kind of combination or strategic development changes. And you can also potentially identify biomarkers that may relate to sensitivity. And so that's the next few slides I want to talk over. So this is something that Neil and I have talked about a lot, and that is, what is it that the mononuclear cells are targeting to release the gamma interferon? And my original belief was it's probably dozens of antigens on the tumor surface. Well, it turns out, now that the technology is getting better to sort out what these markers are, it's hard to do. It costs a lot of money. Only rarely has it been done on patients. But when you look for what's called the driver neoantigen, it typically is 1 or 2 markers on the tumor cell that stimulate the entire immune response against it. I have no idea what makes a particular marker special, but it's typically 1 or 2. Now,
And is it different across
it appears to be different. So that's what's odd, but it's been done so infrequently that we don't know for sure at what level difference. You know what I mean? So let me go into what we know, and Jim may be able to contribute some more on this. The one study that really impressed me in helping connect some of the dots, and before I go into that, let me go back to the neoantigen one. The one thing that I wanted to explain that has surprised me, and you've heard a lot of patients say this, why is it that we're getting this therapy and we have no side effects? Is it really doing anything? And we look at these cells in circulation, and they're very mean cells. When there are some studies, IL-12 is the most obvious one, but the TNF, and when you get these cells in circulation and they release to the wrong target, they cause cytokine storm syndrome, and you die from that. And there are CAR T technologies. So if you mistarget the release of these cells, if these loaded cells with interferon grab an antigen that's a self-antigen, not a tumor antigen, it's going to release the gamma interferon to normal parts of the immune system and cause a lot of side effects, tachycardia, hypotension, a lot of things. So there's different types of antigens. Most are self-antigens on our normal cells, but there are some tumor antigens that are specific to the tumor cell, and there's some tumor antigens that are on cells that aren't like the fetus in the uterus, things like that aren't necessarily active in the body, that are still unique to the tumor, but they're technically not just tumor, you know what I mean? But depending on the timing of the body expression of it, it still may be unique for tumor. So in my thinking, these cells are exquisitely targeting non-self antigens. You get it? Because there's no side effects that we've seen. And so what is there in the literature that shows something similar? There's a neat discussion of several cases where autologous adaptive T-cell transfer, where a group led by a guy named Van der Gaal, I think it was Dana-Farber, took tumors from patients, melanoma patients, expanded them, and then he exposed those tumors to, mononuclear cells from the patients, and then he treated those patients with mononuclear cells, and most of them didn't do that well, but some of them did. Some of them had complete regression of cancer, and 2 patients in particular lived 5 and 9 years without any evidence of disease once they had complete response to their ACT transfer, the autologous adaptive T-cell transfer. So what was special about those patients? And so what he did is he went back and he took the mononuclear cells from those patients 5 and 9 years later. He didn't know who they would be in the beginning, but after they responded, he went back and looked at the reactivity of those patients to their molecular mutated signals. So he took all of the molecular mutations that were identified in these patients from their original tumor, and he created a peptide library. You break apart the proteins that these mutated genes would make, and you expose them to the mononuclear cells these patients had, and you see if they react to any of them. And it's a lot of work. It took a year to do this work. It took $1 million to do it. But he found in patient BO, the longest living one, reactivity to 2 antigens. And in the other patient, AB, he found reactivity to 1 antigen. And he could consistently take their mononuclear cells and get them to release gamma interferon to these peptides. And they wouldn't release to different peptides. You know what I mean? So,
does these 2 patients have anything in common?
No. Melanoma, but there was nothing obvious. So it turns out that somehow their mononuclear cells grabbed the right antigens, what we're terming driver antigens, neoantigens, and they're in circulation, not causing any side effects, but they were deadly to the tumor of these patients. These patients never had recurrence of their cancer. Okay, and so shown on the next slide is the BO was the highlight patient, and he had multiple lesions in his lungs, and they all completely regressed. Now remember the 6 slides I showed you in the beginning when I was saying,
What about the mouse?
what happened to those patients? Meaning there were 6 patients we treated in the clinic, Jim, that had vaccine therapy that we showed in the beginning. You got it now? And so could this mechanism be similar to this mechanism? Now that we've learned more about what's going on. It could. So, there's ways of proving that. So my thought is that the mechanism by which Van der Gaal's therapy worked was he took the peptides that were very similar to the Vigil technology, which was taking the neoantigens. Those peptides stimulated lymphocytes. Those lymphocytes went into the blood and they attacked the tumor. Now he could give these cells, he didn't know that the right cells were selected. He didn't know who was going to get the right ones in the beginning. But after the fact, 9 years later he had to wait. After the fact, he knew that he got something good. And when he went back to look, he found it was to that peptide. He wouldn't have known just like that peptide in the beginning. And guess what? That peptide to AB didn't work. And the AB peptide to BO didn't work. So you don't know in the beginning. So that's a problem of defining the neoantigen. So neoantigen discovery requires both tumor and peripheral blood. Most studies don't store viable tumor. Most studies don't store peripheral blood. Some do, but most don't. So if we go into that, we wanted to verify, so if you look at the other trials are out there, CAR T technologies, other autologous technologies, a variety of antigens have been attempted. We try to guess at what the relevant antigen might be. And in lymphoma, we guessed right with CD19. You'll get up to a 90% response rate in lymphoma patients receiving CAR T to CD19, but you'll have maybe a 10% mortality rate. You still have to go through a transplant to immunosuppress the patient. A lot of toxicity related to it, but that's a pretty active, it's active against the tumor antigen, but also the self-antigen. CD19 is on normal lymphocytes. So that's a problem. So what is it that's unique? Other antigens on there didn't have any activity at all. So when we try to predict what the antigen might be, we're not real good at that. And if we are good at it, we have toxicity.
With that experiment where you did that peptide testing, there were two peptides that were discovered. Was that consistent across different people's tumors?
No.
Well, that's because that's what I kind of assumed. So then the question is, could you have different different drivers in individual cancer?
Exactly.
So no pancreatic cancer could be considered potentially the same. They could have different.
Making conclusions on a couple of patients, and there's a couple of studies that have a couple other patients. You would say that, but we don't know for sure. There could be, like with fusion proteins, there could be 1%, 5%, 10% that are similar.
Right.
You start somewhere and you see how it follows through. But that's right. It may be that it is entirely unique on every patient. It may be related to the MHC process rather than the antigen. We don't know. We don't know.
And that would be the ultimate question, right? It's like having to find which drivers mark each tumor, and how do you do that efficiently? How do you do that?
That's right. That's right. Exactly. So I'm going to tell you. So yeah. As you see. So this is good. So before I go into everything on the next slide. So now we know the elements of defining exactly what you're saying. What is the driver neoantigen? If we can figure that out, we got something. And if we can know what that is, we can expand the cells to above that plateau that we saw the Vigil induce. You can turn them on, but then there's something that causes protected. But if you can expand them ex vivo, you could expand any number you want. You're not limited by the tumor biopsy. You're not limited by how many you can expand. You could give enough. And if it's not causing toxicity, if it's not causing off-target effects, you can expand at a point of, eradicating the tumor maybe. So it needs those. And so to me, before you take the next steps of expensive and creative and first-time ever studies, that's actually what I'm in charge of doing now for this. I'm working with several Nobel Prize guys now on this, so it's kind of cool behind the scenes. I know you're excited about it, but yeah, that's right. So we went to Mount Sinai, which is a premier immune function laboratory. We asked them to verify our samples. To do this work, we're going to have access to a lot of tumor samples, a lot of peripheral blood samples. We have to make sure they're functional. We have to make sure we understand what they are. And so there's a technology called cytop technology where they can take blood cells, flow cytometry label multiple cells at once, and separate each one, and in fact separate each one and identify what they're producing on the inside with respect to cytokines, other peptides, other proteins. Shown here are two patients where we gave Mount Sinai, a patient that we stored samples for 4 years, a patient we stored samples for 3 years, and we asked them to look at the proportion of cells that were CD4 and CD8. We think it's the CD8 cells that are reacting on the lespot assay. That's typically what it is, but we don't know for sure. And when they looked at our peripheral blood cells, they showed that indeed there's a high proportion of CD8 cells. There's a little bit more CD4 cells in the ratio, but there are significant proportions of CD8 cells. And it's relatively similar before Vigil and after Vigil if you look at month 2. And so the ability of those cells in blood doesn't seem to change. Now, their reactivity may change, and that's what the next step is. But if you look at their response to the known positive antigen, that's the PMA. Remember I showed you the PNA and the lespot? They're always positive. Well, these cells are highly reactive to PMA with respect to expressing gamma interferon and TNF. And in fact, the majority of cells express both gamma interferon and TNF. It's not just one expresses gamma interferon, one expresses TNF. So usually when we're getting the cell death to gamma interferon, well, guess what? TNF is also being released, and so it may not be just the gamma interferon that's killing the tumor. And that's helpful for us to know for future development. We can say, well, maybe we should combine with TNF. Well, if we're already getting the TNF expressed by these cells, why add the TNF? So that's something, it's a toxic therapy, why add that? You know what I mean? And so there's a lot of things that have been learned with these results, and it's kind of rapid fire now. On the 23rd, we're going to test where we put tumor cells in there instead of the PMA, and what's going to be reactivity to the tumor cells of the same patient. We'll mix up the tumor cells from different patients, and what's the reactivity to a different tumor. So if you react to the same tumor, you react to a different tumor, and that will help us show, is the antigen different? You can see where these studies go now. And so technically, in all the samples that you guys have done, the majority came from here. You can see that we have 495 untransfected tumor cell samples, 904 transfected tumor cell samples, frozen, 277 paraffin tumor samples, and that'll come relevant a little bit later. Peripheral blood, pre and post Vigil, over 3,000 of these samples, and there's 122 patients. And in the randomized blinded studies, there's another 120 patients today. That's going to grow up to about 230. So you can see we got what we need to figure things out. You're not going to get everything to be positive. You're going to go trial and error, and we're going to lose some samples doing this trial and error stuff. But you can see there's a basis, and we now know that samples out 4 years and 6 years are validated by outside centers, the top immunology centers in the country, and samples internally are verified out 6 months. Tumors, same thing. It's part of product release for FDA. We got to keep stability testing. So, so with that, this is a little bit of what I do behind the scenes. When I'm not here, I'm sort of doing the translational. So I set up a program that basically is going to explore what's going on with the Vigil technology, how can we improve it. We involved immunologists that are world-renowned in helping us with this work. A guy that's actually a knight from Australia, Sir Mark, he's published 862 papers. I mean, he discovered TNF. So, you know, a pretty smart group of guys. The main centers that we're working with are Mount Sinai, Johns Hopkins, and Caris Corporation for something very unique that I'll tell you about. And on an animal model, there are a series of combination technologies that are being looked at based on what we know about this Vigil technology, and we're going to look at expansion studies in that field. We're going to also look at biomarkers, in particular circulating fusion EBS-FOLI. That appears to correlate with disease response in Ewing sarcoma with a center in Germany. So those are things that's the program. If you go to Mount Sinai, this is work being done at Mount Sinai. They're validating the lespot. They're trying to get a more quantitative assay with CyTOF. It's technology I'm not going to go into explain. We're doing the RNA analyses, peptide markers. We're trying to find the neoantigens the same way the Van der Goe guys did it, but the Mount Sinai guys are going to do it the same way. We look at the peptides, make peptide libraries in these patients. So that's already ongoing right now, but it's going to take 6 months to a year to figure out the first patients. But we have patients that are still alive, like I showed you with the bar graphs, out 6 years. They should have died in 6 months. So we have, we think, good cells and a good opportunity to track that. We're going to track the two most dramatic responding patients. That's Carly and Andrew. You guys know them from the Ewing sarcoma work. So that's going on. These are the ELISpot responses of those guys. Carly is funny, I should tell you, because no one else is going to know. We didn't publish this, but she's on the left. She had a good response, and then her ELISpot response started coming down. The bar on the bottom is that 10 level. That's positive. She never went to negative, but you see how she dropped down before the next yellow? We harvested her a second time. She developed recurrent disease about 1.5 years after her first treatment. But we didn't run the ELISpot at the time, but after she had recurrent disease, we ran the ELISpot, and we saw how it dropped down just before her cancer recurred. And so it looks like she was protected for a while, and then it dropped down. One thing that Laura got mad about, her mom, was that her boyfriend apparently was very rude to her and in many ways, and basically she got real stressed with that. Were you there when we were talking to her about that?
I was there when you told me
about
this.
Yeah, and so I don't know, but stress relates to immune system. I'm not making, we didn't say anything about this, but internally, you know, as we deal with our patients, you'd like to know all the things you learn about the research aspects of this, and I, you know, so it was interesting. I hope that didn't contribute in any way, but she's now with no visible disease.
Second dip.
Well, technically the second dip was the boyfriend thing. The first dip was probably the cancer coming back.
Okay.
Yeah, and the second dip was the boyfriend.
Gotcha.
But anyways, so then you want to test this in animal model. So we're looking at, you know, we made a murine plasmid to test a murine, you know, cancer. You have to use the murine cancer, murine plasmid, because the immune system is murine targeting. So our typical xenograft models don't work for evaluation of immunotechnologies, and it's very difficult to evaluate immunotechnologies. So that's work ongoing at Johns Hopkins. This one is the cool one. Jim, you'll like this one. So this is work with Caris, and I'm trying to find a clinically usable way for identifying the driver neoantigen, right? That we're kind of all in agreement. That's the Holy Grail. If we can figure that out, there's a lot we can do with that knowledge in the patient. So the Mount Sinai way, the way Vanderbilt did it, it took a year, and you really got to wait to see the good responding patients, and you might have some. Well, you don't have that luxury when you first treat the patient. You don't know if you're going to be the good responding patient. So, so one of the things that Vigil is associated with, as I showed you, nearly 100% of the patients will turn on this ELISpot response. So the key is they're turning on the ELISpot response. Is the ELISpot response, what is it to? Could it be that there's just not enough cells, and it's actually responding to the right antigen, but there's just not enough cells to control the tumor? If that's the case, what is that antigen, and how could that information help you understand what the antigen is? Well, it turns out that they developed a technology. It's called an aptamer technology. Aptamers are like antibodies, but they're smaller, and they bind to smaller niches. And so tumor cells, when they're in paraffin or they're frozen, do suddenly freeze their MHC expression of their antigens, and when they're thawed, they'll also express it. But the point is that aptamers have an ability to bind to antigen sites. And you can say, well, they bind everywhere. They don't know what the driver antigen is. That doesn't help you any. But if you do what's called a negative selection, if you throw the aptamers onto the tumor, there's a library of 10 to the 15th antimers. I don't even know if that's a trillion or more than a trillion, or if it's infinity. It's kind of like infinity. So they throw a bunch of antimers onto the tumor cells, and then they wash them. And whatever comes back is irrelevant to the cancer. But what stays on is binding to the tumor cells, okay? And you select just the tumor cells to do this on. Then what you do is you take those aptamers and you throw them onto the tumor cells again. And then you throw the lymphocytes that had the ability to release gamma interferon. And what are those lymphocytes going to do to the driver neoantigens? What do you think?
It's going to go in and destroy those cells that are releasing the gamma interferon.
And in part of the destruction process, they have to bind to the neoantigen.
Then you can identify the neoantigen based on what's left.
The way it works, and this is just the technology, when they bind the neoantigen, they'll kick out the aptamer that was binding there. They have better binding affinities than the neoantigen. Neoantigens don't bind. They bind well, but not as good as antibodies and not as good as TCR regions of lymphoid cells. And so when they kick out the aptamer, you capture those aptamers. And once you have those, you can modify their binding affinity. So you modify that, you throw them back onto the tumor cells, and you digest the tumor cells. The aptamers are surrounding the neoantigen, they protect it. So then you harvest the aptamer plus the neoantigen, and you can take to mass spectrometry the peptide sequence of the neoantigen and define it. And that process can be done in 48 hours. So 1 year, 48 hours, which one are you going to take? So this is what we have set up. So this is actually possible to do, we just haven't proven it yet. So this took me about 6 months to set this up. We'll see if we can do it, but everyone's kind of in agreement that it should work. We don't know if paraffin tumor cells will be better or fresh frozen. There's a lot of reasons why plus or minus for either of them, so we're going to run both and compare and see. We're going to look at patients that have lived longer, and the idea is if we can define those patients' neoantigen and they do recur, well then we'll have an opportunity with the knowledge of their neoantigen to take that neoantigen, stimulate their lymphoid cells to expansion, and treat them with a pool of the cells that are targeting that neoantigen, which was relevant before. Make sense?
How do you confirm that the neoantigen?
Mass spectrometry.
Okay.
So it'll tell you the exact wavelength. I don't know what it is. What is the mass spectrometry? But there's a whole plate where you know where peptide groupings will land on based on the mass spectrometry reading, and it's like a little series of waves. And I don't know if it's a frequency or a wave.
All these here,
yeah.
You're making incredibly fine discrimination of different molecular weights.
Yep. If you have 2 nucleotides or 15, or you'll know exactly what the grouping is. So yeah. So anyway, so that's something that over the next maybe just 3 months we may figure out if we can do this or not.
So you're thinking that 48 hours, like, all a patient needs, like, early on, figure it out.
Exactly. So it takes us 1 month with about 80% of the patients to get lespot reactive peripheral blood circulating mononuclear cells. It takes us 2 months to get about 98% of them. It takes about 4 months on the rest, 100%. So I'm saying that if the lespot response correlates with survival, as it seemed to show us way back over here, lespot positive, lespot negative, we're going to be able to gather patients into this group, figure out their neoantigen. It may not be 100% of all patients, but if the technology works, we've already stored the tools to figure that out. We've stored the tumor tissue, we've got paraffin tumor tissue, we've stored the lymphocytes. Run it. Now, we're going to have to optimize it, get it down to a smaller and smaller numbers of tumor cells that are needed to assess this. We're going to get aptamer libraries. We'll have selective aptamer that are relevant to various neoantigens. The ones that work, we'll probably always keep them to rescreen to see if we get a repeat in anyone. So we'll be able to develop things that will make it more efficient over time. But it'll be based on an initial response to lespot, a completion of the Vigil dosing, but then a magnification of the peripheral blood mononuclear cells, specifically the CD8 cells that can target, that have the TCR that can target the neoantigen and give them back to the patient at super high doses. And if it's a non-self antigen, if it's a tumor neoantigen, a driver neoantigen, then that shouldn't have any side effects no matter what dose we use. I mean, obviously, there's doses you get too much, but we'll get there. Does that make sense? So the last slide or two is, when you have a patient and you can potentially track a corollary to the patient's response, you can develop mechanisms with FDA to approve a product before you reach survival. So one of the frustrations I've always had in clinical research is something looks like it's working on a trial, but we can't open it up to the rest of the world because we haven't proven the survival comparative difference. In the Ewings, we've got stuff like that going on. And so if we have a biomarker that correlates with survival, usually you're going to do survival for the first patient group or the second patient group, but in time, if you have a tight correlation between a biomarker and survival, the FDA starts to come around in letting that, based on that information, to letting that be a pre-approval and eventually then a secure approval once the survival is reached, but to let other patients who have that biomarker entered in. So you can imagine if we track the driver neoantigen in circulation, if we know that's the associated relevance to the tumor growth, and we see tumor shrinkage, and we have the driver neoantigen we can measure in circulation, the DNA of it or the peptide of it, and that gets down to 0, and we don't see tumor, that information itself might also help with a FDA reaction to product approval, possibly before you have the end-stage survival. If you get tumor response and correlated driver neoantigens that you're targeting, that ties in mechanism with benefit. You know what I mean? So the Ewings is something we may be able to do that with, and that's why we're looking at for an opportunity for more pressure on FDA for product approval based on a reduction of the circulating EDOS-FLI protein. So that's a call I've got. We've got things set up to do that, get blood samples. We're already storing it the right way. So that's that. There's other things you can do in the last slide for product development, but you get the idea. So that's the value of things going on behind. That's the ongoings of translational behind the scenes, the non-clinical stuff. Cool. Does that make sense? So that's what we're doing.
Very good. I think there's somebody at a group at Washington University St. Louis that has a slightly different approach for neoantigen identification. That is, it's not a year's work. It's a lot of work, but it's into a couple of months. And a couple of steps that are really very useful. It simplifies it in some ways. You might consider, you might want to talk to them and see if you want to have them in parallel.
Yep.
See if their technology and Caris get the same answer. Yep. Or not, it might be interesting. Two things they do is, first of all, you have the list of all the mutations where they have like DNA mutations.
Yeah.
200 mutations.
Yep.
The first thing they do is they look at the transcriptome. And they find that at least 50% of these antigens, for at least 50% of these mutations, there's no RNA.
Right.
So
it's
worthless.
not worth it. Yep.
So they knocked it. So they cut
Yep.
it.
Yep.
Then the next thing they do is simulation. In other words, they look at the predicted sequences. What are the predicted peptide sequences from the remaining group of mutations that are relevant? And then they compare the predicted peptides to the MHC for this patient. So they find that out of these, let's say we're down to 50, that only 3 of them will actually bind to the patient's MHC. So now you're down to this.
Yep.
So that's what they go after. Now you know what you're looking for. And so they managed to winnow this down so you can get to the relevant peptides.
If you've got the paper for their work, grab it to me, because what I'm doing right now is literally flooding those that can do this work and getting samples. Look, we have thousands of samples. Getting samples to them and say, hey, when do you take a shot at 2 or 3 of these? You know.
Okay. I can give you names of the people.
Yeah. Let's.
They're it's a melanoma. Because their last step is make a vaccine.
Yep.
Custom vaccine for that.
Yep. Exactly. So the unique aspect is the lespot response that seems to correlate with survival. That may be an opportunity in that same patient to be able to get the data and literally create the expanded population for them, figure out that peptide early, and then complement in sequence what the Vigil may be doing. It's not good enough. We still don't have a lot of tumor shrinkage. We've got 2 patients that had tumor shrinkage. You know what I mean? That's it's not enough.
I mean, there certainly is existing technology for ex vivo expansion. If you could select the type population. Anyway, I'll find out to send you a paper.
Yeah, that'd be cool. And the CAR-T and the TCR DNA modification approaches, they don't know what to use. They just don't know what the driver new antigen is, so they're having trouble. They're struggling. CD19's worked, but yeah, exactly. Yeah. So.
Interesting. And some of these too, like melanoma, it obviously seems pretty obvious that the immune approach is much more sensitive to certain tumor types than others.
Yeah.
But if you, just because I don't quite understand, like colon doesn't seem to be very immunostimulating, right? It's not really responding to PDLs or any of the old therapies. Do you think that even if you found like a tumor, like the non-kind of immunoresponsive tumors, and you found these presenting cells in a colon patient, theoretically it should work, right?
Yep.
So maybe we just haven't.
There could be tumor microenvironment factors that also have to be overcome. So, in other words, even if you knew that this patient was the colon cancer and the only mutation that's present is any significant cell is KRAS. Okay, so that's what if you found that there are mononuclear cells in this patient that can recognize the mutant KRAS peptide, then why is this not working? And so presumably that's tumor microenvironment factors. So that's why we've got all these studies with the adenosine receptor blockade and all these other kinds of
trying to alter
all these other IDO inhibitors.
Yes, that's
cytokine.
The tumor microenvironment is key. So
the lymphocyte still has to be fully active once it gets to the right place. And there are all these are all reasons why the tumor might be protected, even though there are lymphocytes with specific reactivity are present, but they
are limited by the microenvironment.
Right, they become
inactive. But my strategy is, and it may be wrong, but it's worth trying, I think, is if you take tumor cells, you throw them in a test tube, and you throw 5FU, probably one of our most, well-known chemotherapies, and you just pop it in there, you'll kill every single tumor cell you want with 5FU. But the dose of 5FU needed in various patients is going to suppress the patient, and they're dead there. So there's a dose toxicity limitation. Could it be, if we can construct enough of these mononuclear cells, that we could actually overwhelm the inhibitors? To some degree, to some degree we won't, but you can see where if it's not creating a toxic effect, there's going to be a level at some point of thromboemboli probably, but the point is that you may be able to get enough to overwhelm some of the cross-checks.
I guess
or you do combination technology. Yeah, exactly.
Give a little adenosine receptor inhibitor.
Yep.
I guess my thought on that though would be is that when we looked at the FANG vaccine, for example, we you did it in that one little cohort where we took less tumor, they still had the same response. So more wasn't more.
Yep.
It was less was
The
more.
immune system is tricky because more can sometimes turn on more cross-checks on the immune system. But when you deal with the number of cells that are turned on, it's usually in the production of those cells. Once you have those cells, especially if they're manufactured ex vivo, you can sometimes, the body's not prepared for those cross-checks. It doesn't have a source of viruses that are delivering those cells. You know, they're carrying them over. So it depends on what the technology is. A good start is non-toxic and targeted. You know, that's always been our premise, targeted therapeutics. And this is targeted immune therapies. And this is the ultimate targeted immune. So, yeah. So, cool. Okay. That's it.
Thank you, guys.